Imagine being in an operating room right now. You are standing right over the table, and there are those bright surgical lights glaring down.
Oh, it is such an intense environment to be in.
It really is. And sitting right there in a sterile metal bowl is a human kidney. It is perfectly perfused.
A healthy, vibrant, pristine pink color.
Exactly, pristine pink. And you watch as the transplant surgeon carefully takes that kidney, places it into the patient's abdomen, and meticulously sews the renal artery to the recipient's own blood vessel.
Which is a connection they call an anastomosis in surgery.
Right, an anastomosis. The surgeon is essentially welding two living pipes together, and everything looks completely flawless.
A perfect surgical job.
Yeah. Then the surgeon reaches in and releases the vascular clamp. You watch the recipient's blood rush into the new kidney.
It tumps right up.
It does. It plumps up doing exactly what it's supposed to do. But then within minutes, maybe even seconds, something goes incredibly wrong.
It is a truly chilling scenario to witness firsthand, I have to say. The visual transformation is just jarring.
Because it changes right before your eyes. That perfect pink kidney turns dusky. Then it turns this bruised, terrifying blue.
And finally, it goes completely mottled black.
Yeah. The tissue physically swells up, it completely stops making urine, and it just dies. Right there on the operating table, while an entire surgical team is standing around watching it happen.
Just brutal to watch.
And here's the real kicker. Nobody made a mistake. There was absolutely no surgical error. The plumbing, the stitches, the anastomosis, it was all completely perfect. So what on earth just killed that kidney?
Well, that visceral, almost unbelievable scenario is the core mystery we are tackling in today's deep dive.
Right.
Our mission today is to really unpack the biological mechanics of transplant rejection.
And along with it, the underlying rules of blood compatibility.
Right, exactly. We're going to prove a very specific, overarching thesis to you by the end of this deep dive. The concept is this. Transplant rejection begins as a problem of identity, but it always ends as a problem of blood supply.
I mean, when you think about the phrase organ rejection, or, when you talk about the immune system in general, the imagery that comes to mind is usually a purely invisible microscopic battle.
Right, you picture tiny white blood cells fighting other tiny cells somewhere off in the abstract background of the body.
Exactly. But framing it as a problem of blood supply changes everything. We are talking about a brutal, physical, vascular strangulation.
It's not just cells failing to get along.
No, it is a physical cutoff of the lifeline.
To understand why that pristine kidney died right on the table, to really understand that rapid vascular strangulation, we have to start at the very beginning.
Okay, where do we start?
We have to look at the single, ultimate, existential question that your immune system is obsessed with. The entire architecture of your immunity is built around one endless, repetitive query.
We are talking about the ultimate question of identity, right? Self versus non-self.
That's the one. The immune system basically has one primary job. It goes up to every single cell in your body, and it asks this question millions of times every single second.
Are you me or are you not me?
Exactly. Are you me or are you not me? And it is entirely binary. There is absolutely no gray area here.
So if the cell's answer is me, what happens?
If the answer is me, the immune system ignores it and moves on, just totally leaves it alone. But if the answer is not me, the immune system destroys it.
Just like that. No second chances.
No trial, no deliberation, just immediate execution. When you look at it through that lens, you realize that an organ transplant is the ultimate medical paradox.
Because we are completely violating the most fundamental rule of human biology.
We really are. We are deliberately taking a massive, complex piece of not me, an entire organ from another human being, and surgically sewing it directly into a patient.
And then we spend the rest of that patient's life using complex chemical regimens, immunosuppressants, to desperately try and convince their immune system to ignore this giant, glaring piece of foreign tissue.
But the immune system doesn't want to ignore it. Its entire evolutionary purpose is to find foreign invaders like bacteria or viruses or fungi and eradicate them to keep you alive.
Right. I mean, the immune system doesn't know what a surgeon is.
Exactly. It doesn't know that this new kidney is there to save the patient's life. It only knows that the tissue does not belong.
So basically, our immune system is a hyper-paranoid bouncer checking IDs at the club door. And the club is our body.
That is a perfect way to look at it.
The bouncer is out there millions of times a second, stopping every single cell trying to walk through and demanding, show me your ID. If the ID matches the club's ownership, you go in.
And if it's from out of town, you get tossed out into the alley. Or, well, destroyed.
Right, destroyed. But if the bouncer is demanding to see an ID card, How do the cells actually show it? I'm assuming cells don't have little plastic cards in their wallets. What is the actual physical mechanism of this identity check?
The physical mechanism is rooted deeply in our genetics. To understand how the body prints these ID cards, we really need to trace the blueprint.
Okay, let's trace it.
You start with the foundational rule of genetics. You get one copy of every gene from your mother and one copy from your father. We call each version of a gene an allele.
Okay, so you have a mom version and a dad version. Normally, when we talk about genes, we talk about dominant and recessive traits. Like, if my dad gives me a brown eye gene and my mom gives me a blue eye gene, the brown dominates and the blue is hidden.
Right, that's the classic high school biology example.
So does the immune system pick a favorite parent and hide the other?
Actually, no. The immune system operates under a completely different rule called codominance. For these specific ID card genes, neither allele hides or overpowers the other.
Oh, wow. So they both show up.
Both the mother's version and the father's version are expressed side by side simultaneously. You proudly display both genetic signatures at the exact same time.
Codominance. That makes sense.
And that single concept, codominance, is going to explain almost every weird quirk about blood groups and organ matching that we'll encounter later in this deep dive.
Okay, so if these codominal genes are the blueprint for the ID cards, where's the printing press actually located in our DNA? Like physically, where is it?
The printing press lives on chromosome 6. Specifically, there is a region on the short arm of chromosome 6 called the major histocompatibility complex, or MHC.
Let me break that word down. Histo meaning tissue, and compatibility meaning whether or not things will match and get along. So it's literally the master tissue matching complex.
Yes, exactly. MHC is the general biological term used across all species. Mice have an MHC, dogs have an MHC.
But humans have to be special, right?
We do. In humans, we give this specific genetic cluster a dedicated name. We call it human leukocyte antigen, or HLA.
HLA. Okay, so MHC and HLA are fundamentally the exact same thing in this context.
Absolutely. You can use them completely interchangeably when talking about humans.
Good to know.
Yeah.
So chromosome 6 prints the HLA ID cards. But if I look at most of our human DNA, you and I are essentially identical. I mean, the vast majority of our genetic code is exactly the same.
Right. We share like 99.9% of our DNA.
Exactly. So I'm assuming the HLA genes have to be different. Otherwise, we'd all have the exact same ID card and anyone could donate a kidney to anyone.
You hit the nail on the head. The HLA genes are the most variable genes in the entire human body. There are thousands upon thousands of possible versions or alleles out there in the human population.
Thousands.
Thousands. Almost everyone's ID card is completely and utterly unique to them.
And there's a really fascinating evolutionary reason for that extreme variability. If we all had the exact same HLA genes, the exact same immune ID cards, a single highly adapted virus could figure out the lock, sweep through the population, and wipe out the entire human species.
That is exactly why it exists. The extreme genetic diversity is a defense mechanism. It's what keeps humanity alive against plagues and viruses.
It's incredible. But, wait, let me rephrase that. That variability is a brilliant evolutionary blessing for the species as a whole, but it is an absolute nightmare for transplant medicine.
Oh, it is a total nightmare. Finding a donor whose ID card matches your ID card is incredibly difficult because you are looking for a microscopic needle in a massive global haystack.
It makes total sense. But if it's that chaotic, how do we ever find matches within families? Like, if there are thousands of variations, wouldn't a child's genes just be a random jumble of whatever the parents had?
You would think so, but they aren't a random jumble because of how these specific genes are organized. All of those complex HLA genes sit so incredibly close together on chromosome 6 that they travel together as a single unbroken packet.
An unbroken packet.
Yes. We call this a haplotype. They don't get shuffled individually during reproduction. You get one entire intact packet from your mom and one entire intact packet from your dad.
And because of codominance, you express both intact packets simultaneously.
Precisely. You show both packets.
I picture this like a deck of playing cards. Instead of thinking about genetic inheritance like shuffling a loose deck and dealing out individual cards one by one, it's like your parents hand you a tightly shrink-wrapped pack of cards.
A shrink-wrapped pack. I love that.
Yeah. You get one whole unopened shrink-wrapped pack from mom, and you get one whole unopened shrink-wrapped pack from dad. You don't mix the individual cards up at all.
That analogy perfectly explains the statistics we see at the hospital bedside when we are testing siblings for a kidney donation.
Let's run the sibling math for you, our listener, so you can see how this works in practice.
Think of each parent as holding two distinct, shrink-wrapped packs of cards. Mom has pack number 1 and pack number 2. Dad has pack number 3 and pack number 4.
So every time they have a child, that child gets exactly one pack from mom and one pack from dad. So a child could get pack 1 from mom and pack 3 from dad, or pack 1 and pack 4.
Or they could get pack 2 and 3. Or 2 and 4.
There are literally only 4 possible combinations.
Which means the statistical arithmetic for two siblings sitting in a hospital room is incredibly straightforward. There is exactly a 25% chance, a 1 in 4 chance, that they inherited the exact same two shrink-wrapped packs.
And if that happens, they are a 100% HLA identical perfect match. The holy grail of transplants.
Exactly. And then there's a 50% chance that they share exactly one pack. Maybe they both got pack 1 from mom, but one got pack 3 from dad and the other got pack 4.
So they are half match, which is called haploidentical.
Right, half match. Then, cruelly, there is a 25% chance that they share absolutely nothing. One sibling got packs 1 and 3, the other got packs 2 and 4.
So genetically speaking, when it comes to the immune ID card, they are complete strangers, even though they're full siblings.
Complete strangers to the immune system.
It really demystifies the whole process. It's not esoteric medical magic, it's just basic statistical arithmetic.
Exactly.
So we know how the ID is printed genetically on chromosome 6, but how is it physically displayed out in the world on the cells themselves? Because the body has 2 entirely distinct ways of showing this ID to the immune bouncers.
Yes, in understanding the difference between these two physical displays, the two classes of the HLA card, is perhaps the single most useful concept in this entire field.
Okay, let's break them down.
Let's start with Class I. Class I consists of 3 specific genes: HLA-A, HLA-B, and HLA-C.
A, B, C. Simple enough. The first 3 letters of the alphabet. Where do we find these Class I ID cards?
You find them on the surface of every single nucleated cell in your body. Your skin cells, your liver cells, your heart muscle, your nerve cells.
So basically everywhere.
If a cell has a nucleus, it is wearing a Class I ID card. But there's one major exception to this rule, and it's mature red blood cells.
Wait, mature red blood cells don't have a nucleus?
They actually eject their nucleus during their development. They do it to make more physical room inside the cell to carry oxygen.
That's incredibly efficient.
It is.
Yeah.
But because they have no nucleus, they carry no HLA Class I cards. We really need to remember that exception because it fundamentally changes how blood transfusions work compared to solid organ transplants.
Okay, I'm mentally holding on to the red blood cell exception. So every other cell is holding up this Class I card.
Yeah.
But what is the card actually showing to the bouncer?
The Class I card displays proteins from inside the cell. It's a continuous sampling of the cell's internal manufacturing process. We call these endogenous proteins.
Think of every cell in your body as a little house. The Class I molecule is a front window on that house.
A front window, exactly.
The cell takes a small sample of whatever it happens to be cooking or manufacturing inside, and it puts it in the window display for the whole world to inspect.
And the biological genius of that window system is really profound. Imagine a virus manages to sneak inside the house, infiltrating the cell.
Okay, the virus is in.
The virus takes over the cell's internal machinery and forces it to start manufacturing viral proteins so it can replicate. The cell cannot hide this.
Because of the window?
Exactly. Its internal processing machinery naturally forces those newly minted viral proteins into the Class I window display. The cell is literally forced to confess to the immune system that it has been compromised.
It's basically screaming, look what they're making me cook in here.
Yes. And the immune system has a highly specialized patroller, specifically assigned to look in those Class I windows. It is the CD8 cytotoxic T cell.
The CD8 cells, I get to call them the assassins.
The assassins is a very accurate term. The CD8 cells are the kill squad. They constantly patrol the tissues of your body, looking through everyone's Class I front window.
Just peering in?
The absolute microsecond a CD8 cell looks in a window and sees a foreign viral protein on display, it kills that cell on the spot.
How does it actually do that? Does it just swallow the cell whole?
It uses chemical warfare. The CD8 cell latches onto the infected cell and releases proteins called perforins.
Perforins? That sounds like perforate.
That's exactly what they do. They literally perforate. They punch physical holes in the target cell's outer membrane. Then the CD8 cell drops toxic enzymes called granzymes through those holes.
Wow.
The granzymes trigger a process called apoptosis, essentially forcing the infected cell to commit programmed suicide. The cell implodes, taking the replicating virus down with it.
It's ruthless, but it stops the virus from spreading to the rest of the neighborhood. So that is class 1, the front window showing what's inside, read by the CD8 assassins. Let's look at the second system, class 2.
Class 2 consists of a different set of HLA genes: HLA-DR, HLA-DQ, and HLA-DP. They're the D family of genes.
DR, DQ, DP. Where do these ones live? I'm guessing not on every cell, or we wouldn't need a separate category.
It is highly exclusive. Class 2 is only found on a small, specialized crew of cells known as antigen-presenting cells, or APCs.
Antigen-presenting cells.
The three main professional APCs are dendritic cells, macrophages, and B lymphocytes. Now, activated endothelium, the lining of your blood vessels, can also switch on class 2 when there's intense inflammation, and that plays a huge role in organ rejection later.
Okay, so class 2 is an exclusive card held only by this professional crew. If class I shows what's cooking inside the house, what does class 2 show?
Class 2 shows material that the cell picked up from outside in the environment, exogenous material.
If class on is the front window, class 2 is the neighborhood garbage collector.
That's a great visual.
These antigen-presenting cells, like macrophages, physically roam around the extracellular spaces of your body. When they find garbage, bacteria, shed viral particles, dead cellular debris, they swallow it up.
They do. They chop it into little microscopic pieces inside themselves.
Then they take those pieces and load them onto their class 2 ID card. After that, they act like town criers. They walk right into the local lymph node, holding up this class 2 card with the foreign garbage on it, shouting to everyone, "Look what I found out in the street. Does anyone recognize this? Is this a threat?"
And asking, is this a threat, is the crucial question, because the cell that reads the class 2 card doesn't kill anything directly. The class 2 card is read by the CD4 helper T cell.
The CD4 helper, the commanders.
The commanders. CD4 cells are the generals of the immune army. A CD4 cell looks at what the garbage collector is holding up on the class 2 card. If the CD4 cell decides that the presented material is a genuine foreign threat, it sounds a massive alarm.
It mobilizes the troops.
Exactly. It releases chemical signals called cytokines that mobilize the entire immune infrastructure. It tells B cells to start manufacturing specific antibodies, and it calls in the CD8 assassins to go hunt down anything in the body that looks like that piece of garbage.
It's a beautifully coordinated dual system, front window and garbage collector. To make sure you, our listener, never mix up which T cell reads which class of ID card, there is a brilliant, foolproof mnemonic from the sources called the Rule of 8. It works perfectly every time.
It really is a lifesaver for medical students.
Class 1 multiplied by CD8 equals 8. Class 2 multiplied by CD4 equals 8. They both multiply to 8. 1 is 8, 2 times 4. You can't mess it up.
You really can't. So we have established the architecture of the ID system. We have the genetic passports printed by chromosome 6, and we have the patrollers, the CD8 assassins, and the CD4 commanders.
So what's next?
Now, we need to take all of that biology and put it back into the operating room. Let's look at what actually happens the exact moment a foreign kidney with the wrong ID cards is sewn into a patient.
The outbreak of war.
The outbreak of war. The recipient's immune system, all those patrolling T cells, immediately interact with the newly sewn-in kidney. They read the HLA passports on the surface of the donor organ cells.
And they know instantly.
Instantly. They realize these cards are completely wrong in their shape and structure. This is not self. This moment of recognition is called allure recognition. This declaration of war happens via two distinct pathways: the direct pathway and the indirect pathway.
Let's start with the direct pathway, because when I first read about this in the sources, the mechanism seemed incredibly strange to me.
The direct pathway is explosive, and it is driven by a biological stowaway. When a surgeon transplants a donor organ, they aren't just transplanting kidney cells. Hiding inside the tissues of that donor kidney are the donor's own antigen-presenting cells. We call them passenger leukocytes.
Wait, if these donor cells are just garbage collectors, why are they triggering an attack? Shouldn't they be invisible or even helpful, just cleaning up debris in the new organ?
You would definitely think so. But their presence creates a massive biological short circuit. Those foreign donor APCs eventually migrate out of the transplanted kidney and travel to the recipient's lymph nodes.
Okay, so they leave the kidney.
Right. Now, normally, a recipient's T cell expects to look at its own body's familiar Class 2 card holding a tiny, subtle piece of foreign garbage. But in the direct pathway, the recipient's T cell bumps into this passenger leukocyte.
The stowaway.
Yes. The T cell isn't just seeing a tiny piece of garbage. It is looking at an entirely foreign, intact Class II ID card. The physical shape of the entire molecule is completely alien.
It's like a bouncer expecting to look closely at a driver's license to check a birth date, but instead someone hands them a glowing radioactive brick. The bouncer doesn't even need to read it to know something is catastrophically wrong.
That is a brilliant analogy. The sheer density of foreign HLA on the surface of those passenger leukocytes completely overwhelms the normal lock and key recognition system of the T cells. The reaction is disproportionately massive.
How massive?
Under normal circumstances, during a really bad viral infection, only a tiny fraction of a percent of your T cells will react. But when your T cells encounter intact foreign HLA directly, an astonishing number of them cross-react. Up to 1 in 10 of your total T cells can mount a response.
Wait, 1 in 10. That is a massive percentage of the entire military force deploying all at once.
It is a furious, overwhelming immune storm. This direct pathway is what drives acute cellular rejection. It is the body mounting a massive, fast frontal assault on the organ, sending legions of T cells into the kidney to destroy it.
So if the direct pathway is this massive, immediate explosion that eventually burns out once those passenger leukocytes die off, why do patients still lose organs years later? That brings us to the indirect pathway, right?
Exactly. The indirect pathway is slower, but it never ever stops. Remember the recipient's own garbage collectors, the host macrophages and dendritic cells?
The ones native to the patient.
Yes. In the indirect pathway, the recipient's own APCs are roaming around the new kidney. Now, the kidney cells are naturally shedding bits of their foreign HLA proteins as part of normal cellular turnover.
Just shedding microscopic debris.
Right. The recipient's garbage collectors swallow up those shed foreign ID cards, chop them up, and present those tiny pieces on their own familiar Class II cards.
So they're going back to the lymph nodes going, look at this weird foreign plastic I found in the neighborhood.
Precisely. They take it to the CD4 commanders. The commanders recognize the chopped up debris as foreign garbage and start coordinating an attack.
So how does this differ in speed from the direct pathway?
Because it requires swallowing, processing, and presenting shed garbage, the response is much quieter and slower than the direct pathway. However, as long as that foreign organ is in the body, it is shedding foreign HLA.
So it never ends?
Never. The indirect pathway is a slow, grinding war of attrition that drives chronic rejection over months and years.
The direct pathway is the massive explosion of acute rejection, and the indirect pathway is the relentless grinding of chronic rejection. But either way, the recipient's immune system has read a foreign passport and declared war.
A war that must be fought on a physical battlefield.
Right. Now, returning to the core thesis of our deep dive. We know the immune system is angry. We know it has declared war. But where is the physical damage actually happening? An angry CD4 cell doesn't just magic an organ to death. It has to physically dismantle it.
The physical battlefield where the destruction occurs is the endothelium, which is the inner lining of the blood vessels.
But why the blood vessels specifically? Why doesn't the immune system just, chew on the outside of the kidney?
Because the endothelium is the very first piece of donor tissue that the recipient's blood actually touches. Think back to the operating room from our intro.
The moment they release the clamp?
Yes. The instant that vascular clamp is released, the recipient's blood, full of billions of immune patrollers and circulating antibodies, comes rushing forcefully into direct contact with the inner walls of the donor's blood vessels. It is the unavoidable front line of the war.
So it all starts at the vessel wall.
It does. We can categorize this vascular destruction into 3 distinct time frames: hyperacute, acute, and chronic. Let's look at hyperacute rejection first. This is the phenomenon of the kidney turning black on the table in minutes to hours.
I still struggle to understand how it can happen so fast. Even the direct pathway takes days or a couple of weeks to really ramp up a T-cell response. How does a kidney die in 5 minutes?
It happens instantly because the immune army doesn't have to take the time to mobilize. It is already fully armed and waiting. Hyperacute rejection occurs only when the recipient already has preformed antibodies waiting in their blood before the surgery even starts.
Preforms antibodies, so the bodyguards are already heavily armed and patrolling. But why would a patient already have antibodies against a kidney they've never seen before?
It usually happens through previous medical events. A patient can develop anti-HLA antibodies from a previous failed organ transplant or from prior blood transfusions.
Because transfused blood has white blood cells in it, right?
Exactly, and those white cells carry foreign HLA, or they can develop them from previous pregnancies. During pregnancy, the mother is naturally exposed to the father's foreign HLA through the baby's shed cells, and her immune system builds antibodies against those specific paternal HLA markers.
So they have this massive standing army of preformed antibodies just circulating in the plasma, doing absolutely nothing until the exact moment the surgeon releases the clamp.
The blood rushes in. Those preformed antibodies immediately recognize the foreign HLA covering the lining of the donor's blood vessels. They bind to the endothelium by the millions.
And what does that binding actually do?
This binding triggers a system called the complement cascade.
The complement cascade, what actually is that? It sounds like an add-on, but it's incredibly destructive, isn't it?
Oh, it is devastating. Complement is a cascade of proteins naturally circulating in your blood. When antibodies bind to a target, they act like a homing beacon that activates these complement proteins.
A homing beacon for destruction.
Yes. The proteins assemble themselves into a microscopic cylinder that physically punches a massive hole in the cell membrane. But worse than that, complement activation triggers immediate massive blood clotting.
So the antibodies bind, the complement punches holes, and the blood instantly clots.
Instantly. Every capillary, every microscopic vein inside the kidney thromboses. The vessels completely clog with clots. The organ suffers a massive infarct tissue death due to a lack of oxygen, which we call ischemia.
It is a pure vascular catastrophe.
It suffocates. That is why it swells and turns mottled black on the table.
You can't reverse this once it starts, right? You just have to take the kidney back out.
You have to remove it immediately to save the patient. There's no coming back from it. This is why we do extensive matching before surgery to guarantee a hyperacute rejection cannot happen.
So that's the hyperacute nightmare. Let's move to the second time frame, acute rejection. This happens over days to months.
Right, and this is driven by that furious direct pathway we talked about, the massive T-cell response to the stowaway APCs.
Since the vessels aren't clotting instantly like in hyperacute rejection, what is the actual damage looking like in acute rejection?
Acute rejection comes in two flavors, but both relentlessly attack the vessels. First, there's acute cellular rejection. Those mobilized CD8 assassins and CD4 commanders infiltrate the organ and physically attack the cells, particularly the walls of the blood vessels.
So they attack the pipe itself.
Yes. This causes severe inflammation and swelling of the vessels, a condition called endotheliolitis. The vessels get so swollen that blood struggles to get through.
And the second flavor?
The second flavor of acute rejection is antibody-mediated. The immune system has now had a few weeks to manufacture brand new antibodies, specifically targeting the donor's HLA.
So these are fresh recruits, not a standing army.
Exactly. These newly minted antibodies rain down on the blood vessel lining. And in pathology, if we take a needle biopsy of the struggling kidney, we can actually see the physical footprint of this antibody attack.
How do you see it?
We use a special chemical stain called a C4T stain. C4T is a degraded fragment of one of those complement proteins we just talked about. When an antibody activates complement, the C4D fragment gets covalently bonded to the capillary wall.
It stays there like a permanent scar.
It does. When we see it lighting up under the microscope, we know definitively that antibodies are dropping bombs on the blood supply.
Whether it's T cells causing severe inflammation or new antibodies triggering complement bombs, the diagnostic marker of the acute attack is always found in the blood vessel.
Always. The vessels are the target. Finally, we have chronic rejection. This happens over months to years. This is driven by the slow, relentless, indirect pathway. The garbage collector is constantly showing the commanders shed pieces of the organ, decade after decade.
So what happens to a blood vessel when it is subjected to a slow, relentless, low-grade immune attack for years? I mean, it doesn't clot, it doesn't violently swell, so how does it fail?
The vessel responds in the only biological way it knows how to defend itself against constant injury. The inner wall of the blood vessel, called the intima, starts to physically thicken.
It gets thicker.
The cells proliferate, laying down scar tissue. As the vessel wall gets thicker and thicker, the opening inside the pipe, the lumen, gets narrower and narrower.
It is exactly like rust slowly building up inside an old iron plumbing pipe. The water pressure drops over years until it's completely occluded.
The medical term for this is transplant vasculopathy, or graft arteriosclerosis. The pipe slowly closes up, the organ is progressively strangled, chronically deprived of its life-giving blood supply.
And the end result?
Slow ischemia, followed by fibrosis, scarring, and the gradual death of the organ's functional tissue. It just withers away.
Let's look at the whole arc we just painted. Hyperacute rejection, the vessels instantly clot. Acute rejection, the vessels are inflamed and antibody bombed. Chronic rejection, the vessels slowly rust shut and strangle the organ.
That's a full spectrum.
It completely changes how you view a transplant failure. It is not the kidney tissue inherently forgetting how to filter urine. It is the kidney being actively starved to death by the systematic destruction of its blood supply.
Yes. Underneath all the complex immunology, transplant rejection is fundamentally a vascular disease wearing an immunological mask.
Vascular disease wearing an immunological mask, that's powerful.
That is the unifying truth of the entire field of transplant medicine. Once you grasp that concept, every other detail makes perfect sense. And the brilliant transition here is that if solid organs are essentially just vast networks of blood vessels, what happens when the transplant itself is the blood?
Right, because a blood transfusion is technically a liquid tissue transplant. You are putting foreign human tissue into a patient's veins.
Yes, you are. And the logic for blood transfusions is exactly the same biological logic we just used for solid organs. It's just a vastly simpler system. If you understood everything we just discussed about transplant rejection, understanding blood compatibility is going to be incredibly intuitive.
So we are diving into the ABO blood group system. Forget the heavy medical terminology for a moment. Let's visualize every single red blood cell in your body as a tiny little sphere. And on the surface of this sphere, the cell can fly little flags to identify itself.
Okay, I'm picturing it.
In the ABO system, there are only two possible flags, an A flag and a B flag.
Just two flags. If your red blood cells are flying the A flag, you have group A blood. If your cells are flying the B flag, you have group B blood.
And because of that concept we discussed at the very beginning, codominance, where genes express simultaneously without hiding each other, if you inherit an A from one parent and a B from the other, your cells fly both flags at the exact same time.
You proudly fly both flags, so you have group AB blood.
And finally, what if you don't fly any flags at all? We call that group O. The easiest mental trick to remember this is to visualize the letter O as a zero. Zero flags on the surface.
So the flag system is simple. A, B, both, or zero. But what about the immune system's response to these flags?
This is where the immune bodyguards come in. Your blood plasma is constantly patrolled by bodyguards, which are antibodies.
And their rule of engagement is brutally simple. They will violently attack any flag that you do not personally wear on your own cells.
Let me walk through that. If I am group A, I wear the A flag. That means my bodyguards look at the B flag and immediately recognize it as foreign. So group A plasma is full of anti-B bodyguards.
Correct. If you are group B, you wear the B flag. Your bodyguards hate the A flag. So group B has anti-A bodyguards. Now what happens if you are group AB? You wear both the A flag and the B flag.
Since they wear both flags, they can't possibly have bodyguards against either one, otherwise they would be constantly attacking their own blood. So group AB has absolutely zero ABO bodyguards in their plasma.
And because they have no bodyguards to attack incoming blood, group AB is the universal recipient. You can transfuse them with A, B, AB, or O blood, and they will accept all of it peacefully.
Then we have group O zero flags. They don't wear A, and they don't wear B. Because their cells wear no flags, their immune system considers both the A and B flags to be highly dangerous foreign invaders.
So Group O has both bodyguards, anti-A and anti-B, permanently patrolling their plasma.
But the flip side of that is why Group O is so incredibly valuable. Because Group O's own red blood cells carry zero flags on their surface, they are essentially invisible to anyone else's bodyguards.
They're stealth cells.
Exactly. You can pump Group O red blood cells into a person with Group A, Group B, or Group AB blood, and those patients' bodyguards will have absolutely nothing to grab onto. That is why Group O is the universal donor.
That is the mechanical logic of universal donors and recipients. But we need to address the most critical, often missed point about the ABO system. We need to talk about where these bodyguards actually come from.
Right. Earlier, we established that the immune system usually only builds antibodies after you're exposed to a threat, a recruited army. So how does a person develop anti-A or anti-B antibodies if they've never had a blood transfusion in their life?
This is the vital difference. The ABO bodyguards are a permanent standing army. You do not need to have a mismatched blood transfusion to develop them. You are born developing them from infancy.
From infancy, how?
Due to an evolutionary quirk called molecular mimicry.
Molecular mimicry. What is mimicking what?
When a baby is born, their digestive tract is rapidly colonized by ordinary healthy gut bacteria.
Yeah.
It just so happens that the surface sugars on the cell walls of certain common intestinal bacteria are structurally almost identical to the human A and B blood group flags.
Wow.
The infant's immune system recognizes the bacteria as non-self and mounts a defense. But in doing so, it inadvertently builds a massive, permanent standing army of anti-A and anti-B bodyguards that cross-react with human blood groups. They are permanently armed and on guard for the rest of your life.
Why on earth would gut bacteria look like blood cells?
It's just a biological coincidence of molecular shapes. Sugars can only assemble in so many ways. But the consequence is massive, because that standing army is always there, fully armed and waiting. A wrong blood group transfusion is an immediate, lethal catastrophe.
If you accidentally transfuse Group A blood into a Group O patient, you are pouring millions of A-flag cells directly into a patient whose massive standing army of anti-A bodyguards is already waiting.
The antibodies attack immediately. They bind to the transfused cells, activate the complement cascade we talked about earlier, and punch holes in the red blood cells.
The cells literally explode right inside the blood vessels.
Yes, this is called intravascular hemolysis, and it can throw a patient into shock and kill them in minutes.
Notice how perfectly this connects back to the solid organ transplant. This is the exact same underlying mechanism as the hyperacute organ rejection we saw in the cold open.
It absolutely is.
Preformed antibody, immediate complement activation, instant vascular destruction. Whether it's a kidney dying on the table or a bag of mismatched blood exploding in the veins, it is one disease mechanism manifesting in two different ways.
The underlying pathophysiology is completely identical. To tie the genetics of ABO together, let's use an analogy to visualize how a gene can be broken. We know the ABO gene lives on chromosome 9. We know A and B are codominant. But what exactly is the O gene?
O is a recessive trait, right?
Yes, O is actually a recessive trait, a genetic mutation that breaks the system.
I love the cupcake analogy for this from our sources. Think of the basic immature red blood cell structure as a plain, undecorated cupcake. In genetics, this plain base is called the H antigen.
It's the structural foundation of the cell.
Right. Now imagine the A gene and the B gene are tiny little robotic decorating machines inside the cell. If you inherited a working A gene machine, it runs around dropping a specific A topping onto the plain cupcake.
And if you inherited a working B gene machine, it drops a different B topping onto the cupcake.
And if you have both machines, because of codominance, both machines work furiously at the exact same time, and your cellular cupcake gets covered in both the A topping and the B topping, Group AB.
So what is the O gene? The O gene is simply a broken decorating machine. It is a mutation that produces an enzyme that lacks the ability to attach any toppings. It's essentially switched off.
So it does nothing.
Right. If you only have O machines, they run around doing absolutely nothing. The cupcake stays plain. That plain cupcake is your zero flag Group O red blood cell.
This explains recessiveness perfectly. If you inherit an A machine from your mom and a broken O machine from your dad, your genotype is AO. But because you have at least one working A machine, it's going to run around and successfully put the A topping on every single cupcake.
The working machine completely masks the broken machine.
Exactly. To actually be Group O blood, you have to inherit two broken O machines, one from mom, one from dad. Two broken machines mean the cupcake stays plain forever.
It perfectly demonstrates codominance and recessiveness at the cellular level. Now, we have one more crucial piece of the blood puzzle. There is a second flag system layered directly on top of the ABO system.
When people say I'm A positive or I'm O negative, that positive or negative refers to the Rh factor.
Yes, the Rh factor, specifically known in the lab as the D antigen, it is encoded on chromosome 1. The rules are incredibly simple. You either carry the D flag, which makes you Rh positive, or you lack the D flag entirely, making you Rh negative.
So it's just another flag. But there is one vital, absolutely critical difference between the ABO flags and the Rh flag. And this difference changes how hospitals handle blood matching entirely.
It is the most important distinction in modern blood banking.
Yeah.
Unlike the ABO system, Rh negative people do not have a preformed standing army of bodyguards.
Wait, if I am O negative, I don't naturally just have anti-Rh antibodies floating around from gut bacteria?
You do not. The gut bacteria do not mimic the Rh flag. The anti-D army must be recruited. It only appears after your immune system is physically exposed to D-positive blood for the very first time. In medicine, we call this first exposure sensitization.
Sensitization. Okay, let me deduce what happens here. If an Rh negative person is accidentally given a unit of Rh positive blood for the first time, I'm assuming nothing terrible happens immediately because there are no bodyguards waiting to attack it.
Your deduction is spot on. The first time, there is very little drama. The immune system slowly recognizes the foreign D flags floating around, realizes they don't belong, and spends the next few weeks slowly recruiting and building an anti-D army.
What happens to the transfused blood?
The transfused cells eventually die off naturally. But
The second time is the trap.
the second time is incredibly dangerous. Because now the army has been fully recruited. It have immunological memory. It is waiting. If you were exposed to Rh-positive blood a second time, that recruited army attacks with massive destructive force.
It's lying in wait.
Yes. This first time, okay, second time, dangerous behavior perfectly mirrors how HLA antibodies cause acute and chronic rejection in solid organ transplants. It's a recruited army, not a standing army.
Where this recruited army concept truly becomes a matter of life and death is in obstetrics. Let's talk about hemolytic disease of the newborn.
This condition illustrates the stakes of sensitization perfectly. Imagine an Rh-negative mother is pregnant. The father is Rh-positive, and the baby growing inside her inherits the father's genes and is Rh-positive.
The baby is flying the D flags, but the mother is completely lacking them.
During the first pregnancy, everything is almost always fine. The placenta keeps the mother's blood supply and the baby's blood supply physically separate. But at the moment of delivery, as the placenta detaches, there is an inevitable mixing of blood.
And the leak happens.
Small amounts of the baby's Rh-positive red cells leak directly into the mother's maternal circulation.
The mother has just been sensitized.
Her immune system sees those foreign D flags from the baby's leaked cells. Over the next few weeks, she recruits and builds a permanent, powerful army of anti-D antibodies. She's now heavily armed against the Rh flag.
Which sets the trap for the next pregnancy.
If she gets pregnant again with another Rh-positive baby, she now has this standing army of anti-D antibodies. And crucially, these specific recruited antibodies are a molecular class called IgG. IgG antibodies are physically small enough to cross the placental barrier.
Unlike the ABO antibodies, which I'm assuming are too big.
Actually, yes, you are correct. ABO antibodies are mostly a class called IgM, which are massive, snowflake-like structures that cannot cross the placenta. But the Rh antibodies, the IgG, pour easily across the placenta, enter the developing fetus's circulation, and begin systematically destroying the baby's red blood cells while it is still in the womb.
That is devastating.
The fetus suffers massive anemia and heart failure. It is devastating.
The medical fix for this is honestly one of the most elegant interventions in human history. Can you explain the ROGAM shot? Because the logic of how it works is so incredibly clever. We inject the mother with the very thing we're afraid she's going to make.
It is a stroke of biological brilliance. ROGAM is essentially a highly concentrated dose of manufactured anti-D antibodies, immunoglobulin. We give this intramuscular injection to the Rh-negative mother at specific times during her pregnancy and immediately after delivery.
We deploy a temporary mercenary force.
A mercenary force, exactly.
When those few stray Rh-positive baby cells leak into the mother's blood at delivery, the ROGAM antibodies we injected instantly hunt them down, bind to them, and completely mop them up before the mother's own immune system ever gets a chance to see them.
We artificially mask the foreign antigen. Because our immune system never actually sees the foreign D flags, it never gets triggered. It never recruits the permanent anti-antibody army, and she never builds that deadly immune memory.
It's like sweeping the footprints away before the guard dog wakes up.
Exactly. We prevent the sensitization process entirely. Because of this single injection, the next pregnancy is completely safe. It has saved millions of lives.
Millions. It's amazing. We have covered the ABO standing army and the Rh recruited army. Because the stakes are so incredibly high, whether we are talking about a full kidney transplant or a simple bag of blood, we can never just guess.
No, we absolutely cannot guess.
We can never just cross our fingers and hope we got the math right. We have to do a dress rehearsal.
We must do a dress rehearsal in a test tube before we ever touch the patient. In the lab, this is called cross-matching. We take the donor's blood and the recipient's blood, and we physically mix them together to see if a microscopic war breaks out.
And there are two types, right?
There are two distinct types of cross-matches: the major cross-match and the minor cross-match.
Keeping these two straight can be confusing, but the trick is asking one simple question. Whose army is meeting whose flags?
Let's break down the major cross-match first. In a major cross-match, we take the donor's red blood cells, the incoming flags, and we mix them with the recipient's serum, which is the liquid part of the blood that holds the antibody bodyguards.
We are checking if the recipient's bodyguards will attack the incoming donor cells.
Yes. And it is called a major cross-match because the recipient is the host. The host's immune system is massive. It is the major power in the room. They are on their home turf with their entire standing army, and the donor cells are just a tiny, outnumbered incoming force.
So if there's a reaction,
If the major cross-match is positive, meaning the recipient's antibodies violently destroy the donor cells in the test tube, you absolutely cannot give that blood. It will cause a catastrophic, fatal reaction in the body.
the host is the boss. The recipient's immune system dictates everything. That is the major threat. What about the minor crossmatch?
In the minor cross-match, we flip the components. We take the recipient's red blood cells and mix them with the donor's serum. We are checking if the tiny amount of antibody bodyguards floating in that one single bag of donor plasma will attack the recipient's massive bodily supply of red blood cells.
Why is that considered minor? A bodyguard is a bodyguard, right? Wouldn't they still attack?
It's considered minor purely because of volume and dilution. When you transfuse that bag of blood into the patient, the relatively small amount of donor antibodies gets rapidly and massively diluted in the recipient's huge total blood volume.
No, I see. It just gets watered down.
Right. It might cause a tiny bit of red cell destruction, but it is vastly less dangerous than the major cross-match scenario where the entire host army attacks the incoming cells. The guest army is a minor threat.
Host army equals major threat. Guest army equals minor threat. What I love is the beautiful symmetry here. This cross-match logic for blood is exactly the same logic used for solid organs.
It translates perfectly.
Before a kidney transplant, they take the recipient's serum and test it against the donor's kidney cells. If that test is positive, it means the recipient's bodyguards are already heavily armed against the kidney.
It means you have just perfectly predicted a hyperacute rejection in a test tube. You have watched the catastrophe unfold before it actually happens in the operating room.
So it's a huge red flag.
That positive cross-match offers a life-saving no. You cancel the surgery and look for another donor.
What happens if something slips through the cracks? Despite all these elaborate checks, mistakes happen. Usually it's a clerical error, like someone putting the wrong patient sticker on a blood bag. The sources outline a few types of transfusion reactions when a mismatch occurs. Let's run through them.
The most feared is the acute hemolytic reaction. This is the ABO disaster we discussed. The recipient's massive standing army instantly destroys the donor cells right inside the blood vessels.
What does that look like for the patient?
The clinical presentation is dramatic. The patient develops a sudden, spiking fever, severe lower back or flank pain as the kidneys clog with cellular debris, their urine turns dark red or brown from the destroyed red cells, and they can rapidly crash into shock. This is the one that kills rapidly if not stopped.
And again, perfectly preventable just by meticulously checking the name on the bag at the bedside. What are the other reactions?
You might see a febrile non-hemolytic reaction. This is actually quite common and generally mild. The recipient has pre-existing antibodies against the donor's white blood cells that snuck into the bag, not the red cells. The attack releases inflammatory cytokines, causing a fever and chills. It's uncomfortable, but it's not deadly.
Then there are allergic or anaphylactic reactions where the recipient's immune system reacts to random proteins floating in the donor's plasma. You get hives, itching, or in severe cases, airway swelling.
Finally, we have the delayed hemolytic reaction. Remember our recruited army? This happens when a patient is exposed to a minor blood antigen they were previously sensitized to, perhaps years ago from a prior transfusion.
So the body forgot about it.
Over time, their antibody levels dropped so low that they didn't even register on the initial lab cross-match. The blood looked perfectly safe.
But the new exposure wakes the immune system up.
Exactly. The immune memory cells recognize the antigen, rapidly recruit a massive new army of antibodies, and days or even weeks later, they slowly start destroying the transfused blood. The patient might just feel fatigued or mildly jaundiced a week after going home.
The family resemblance across all of this is undeniable. A preformed antibody, like in the ABO system or hyperacute organ rejection, gives you an immediate explosive vascular catastrophe. A recruited antibody, like in Rh disease or delayed transfusion reactions, gives you a delayed building response.
It's a great summary.
One single physiological sentence basically covers hyperacute rejection, acute hemolytic reactions, Rh disease, and delayed transfusion reactions all at once.
It is all the exact same fundamental physiological mechanism, just expressing itself on different timelines, driven by different immune triggers and attacking different tissues.
We have covered an immense amount of ground today, from cellular checkpoints to operating rooms to maternity wards. Let's try to collapse this massive deep dive of immunology and genetics down into the core takeaways that our listeners now permanently own.
I could summarize it in a few key pillars. First, remember that the immune system only ever asks one single question, self or non-self. The answer to that question is physically written on HLA ID cards.
The passports.
Yes. Those cards are encoded on chromosome 6, they are inherited in unbroken haplotype blocks from your parents, and they are expressed codominantly, meaning you show both your parents' cards at the same time without hiding either.
Pillar 2 is the rule of 8, which explains how those cards are read by the immune bouncers. Class I cards are on every nucleated cell, they show what's cooking inside the cell, and they are read by the CD8 assassins. 1 times 8 is 8.
And Class 2 cards are exclusive to the garbage-collecting APCs, they show what's found outside the cell, and they are read by the CD4 commanders. 2 times 4 is 8.
Pillar 3 brings us back to our central thesis. Rejection is fundamentally an identity problem, but it always cashes out at the endothelium.
Whether it is immediate clotting from complement activation, acute inflammation from T cells, or chronic slow strangulation from rust-like thickening of the vessel walls, the transplanted graft ultimately dies because its blood supply is attacked and destroyed.
Rejection is a vascular disease wearing an immunological mask.
Pillar 4, blood, is the exact same story, just simplified to a few basic flags. The ABO system relies on a permanent, preformed, standing army of bodyguards triggered by molecular mimicry from gut bacteria.
Which is why a mismatch causes an immediate intravascular disaster.
Exactly. But the RH system and the HLA system for solid organs rely on a recruited army. They are usually safe the first time you see them, but incredibly dangerous once you are sensitized.
Finally, when you are trying to understand a cross-match in the lab, always remember, the host is the boss. The major cross-match tests the recipient's powerful antibodies against the donor's cells because the recipient's massive immune system rules the room.
Identity decides whether the war starts, but the blood vessels decide exactly how the organ dies.
Identity decides whether the war starts. The vessels decide how the organ dies. That is a framing that will stick with me for a long time. It clarifies so much chaos. Before we wrap up, I want to leave you, the listener, with one final, deeply provocative paradox to ponder on your own. It's a thought that builds on everything we've just discussed about the origins of this system.
It's a fascinating paradox.
We learned earlier that the extreme, chaotic, genetic variety of the HLA ID cards evolved as a brilliant, necessary defense mechanism. It guarantees that no single virus or plague can easily figure out the universal biological lock and wipe out the entire human race in one fell swoop. Our survival demands this diversity.
It's what keeps us here.
But think about the profound biological tragedy of what that actually means. The exact evolutionary trait that guarantees the survival of humanity as a vast collective species, our staggering infinite immune diversity, is the very same trait that makes it incredibly agonizingly difficult for us to save a single individual human life through transplantation.
It's the ultimate double-edged sword.
Our bodies have been perfectly honed over millions of years to survive plagues. But in doing so, they have become perfectly evolved to violently reject salvation from a modern surgeon's scalpel.